Thanks everyone for coming along. Very nice to see this great turnout. I don't know if you knew, I don't really like giving talks, so I hope it'll go well. As Chrissy said, and as many of you know, I am a core scientist here, and I'm a core scientist in the Highlander Lab. So here is an overview of people who are part of the Highlander Lab and affiliated with the Highlander Lab. And most of you will know that our chief Highlander is Gregor Goyant. And in the Highlander Lab, we do breeding, we work on breeding, we do quantitative genetics. So we bring together data science, genetic, and breeding research. And we work on a wide variety of systems, many animals, but also plants. And insects, which don't count as animals for this talk here. Okay. About myself, Chrissy said that I have a background in biology, quite a bit of molecular biology, but then from PhD times, I moved into computational biology, and I would say I'm a computational biologist now. Okay, so we are in the age of whole genome sequencing now, and it's quite easy to generate whole genome sequencing data sets for individuals of most species. It's not incredibly expensive anymore. It's cheaper than paying a postdoc for a month, for instance. And we have many of these data sets rolling around. And common questions with whole genome sequencing data sets are, what is the genetic diversity in that individual that I just sequenced? What is his heterozygosity? And then usually you can also extrapolate from looking at one individual. You can say what is likely the population's genetic diversity. You may ask, if I have this genome sequencing data set, what is actually the genome size of that individual that I just sequenced there? Or if you have already made a genome assembly, you can ask, is this genome assembly complete? You can ask, what's the ploidy level of the individual that I just sequenced? For some, that will not matter because they work on diploids, but many people who work on other systems have actually mixed ploidy species, and it can be quite interesting to know whether something is diploid or polyploid. If you know you have a polyploid, you may be interested in whether there is some divergence between the subgenomes, or whether they are all equivalent. And then finally, commonly asked question is whether there are any contaminations in our whole genome sequencing dataset. And all these questions can be answered with k-mer methods. And that's what I want to talk about today. And using k-mer methods is also often called genome profiling. So genome profiling is the assessment of genome properties from sequencing data without using a genome assembly. So you have a pile of sequencing reads, you try to draw conclusions from that without mapping them to a reference or without generating a genome assembly. And so this is the computational genetics discussion group, and it would be nice if this was a bit interactive. So please interrupt me and ask questions if something's unclear, or if you feel you want to say something.
Sorry, can you repeat your definition on genome profiling?
Genome profiling. I should have written it out. It's the assessment of genome properties by looking only at the sequencing reads. So if you do whole genome sequencing, that's nowadays done in a way that's called shotgun sequencing. You sequence random bits of the genome, and then you have a whole pile of reads. And usually to make sense of these, you need to take these sequencing reads and map them to a reference, call variants, ask where are the heterozygous sites. Or if you don't have a reference, you even have to go and assemble these reads first. So genome profiling is trying to draw conclusions in the absence of a genome assembly.
My understanding was that to just even generating the sequencing, you need to have the reference.
Yeah. No, you just need DNA and you put it in a machine and oops, your uncle.
So is it a BAM file basically working with?
Well, any file that can contain reads, so it might be FASTQ or FASTA or BAM. Okay, thank you for asking. So here's the outline. we start with an introduction to give you a bit of an intuition about k-mers. What are k-mers? What is a k-mer spectrum? Then we think about how to model a k-mer spectrum, the shape of a k-mer spectrum. Then I'll talk about applications, some work that I've done and some new stuff that I'm working on. And then there'll be a summary at the end. Introduction. So building an intuition for k-mers. What actually are these k-mers? Well, a k-mer is a short substring of a defined length, k, of some sequence. And in the context of genomics, these sequences are genome sequences or sequencing reads that we created from genomes of organisms. And you can pick any size that you want. For instance, you can make k-mers of length 3 from our sequence here. And then you can chop the sequence into overlapping k-mers. And unsurprisingly, the type of alphabet that you use in DNA, it's only A, C, T, and G, so there are 4 letters. And the k-mer length then lets you easily conclude how many possible individual k-mers there are. So if we have a k-mer length of 3, there are only very few different k-mers possible, and you get easily k-mers that are identical by chance. Can anybody see a couple of identical k-mers here? That's correct. Thank you. Some audience participation. That's good. So these are identical. And we usually don't want that, so we make the k-mers longer. And there is a perhaps unsurprising relationship between the value of k, the k-mer length, and the number of unique k-mers that are possible. So the y-axis here is logarithmic. And you can see that if you pick a k of 21, you make 21 mers, then you get so many different k-mers that you have more than even the largest genomes that we know. So 21 and 27 are quite popular values. This is also the length of a PCR primer. So how can we summarize this k-mer information? We have a bunch of sequencing reads. Oops, sorry. Turn it into k-mers. Well, maybe a very basic way is to write them all out in a very long text file. So we have here a species, Euphrasia roscoviana. It's got a genome size of 600 megabase pairs. And in that sequencing data set, we found about 400 million individual k-mers. And they're listed all here. So this is a truncated printout. And you can see the sequence composition of the k-mer. And then you can see how many times that k-mer was observed. And that's not the most useful way of representing such a data set, but you can add a second round of counting and then ask how many k-mers are there that are observed exactly 119 times, and how many k-mers are there that are observed exactly 12 times. And that is what is called a k-mer spectrum. And they are often produced by people who are interested in heterozygosity and in genome completeness. Often if you read a genome sequencing paper, if you don't find this sort of thing too boring, then you find in the supplemental information at least one k-mer spectrum. So summarizing k-mer information that can be done in a k-mer spectrum. K-mer datasets are very large. So these k-mer spectra here are a very small summary of the k-mer dataset. Don't get confused. A k-mer spectrum shows on both axes counts, right? These counts here on the x-axis, they're called multiplicity. So this is how many copies of some identical k-mer are there. And then on the y-axis, we say how many k-mers are there that are observed exactly 100 times. And these counts on the y-axis, they get quite high, into the millions. You can also show that as a density, of course, if you want to. So the k-mer spectrum is a summary of sequencing data set. The plural of spectrum is spectra, 1 k-mer spectrum, 2 k-mer spectra. And generally, they are generated from sequencing data, sequencing reads, not so often from genome assemblies.
And sorry, why would do we not get the monotonic trend here?
Aha, we'll come to this in a second. Yes. Modeling k-mer spectra. right? So the takeaway from this, if you don't take anything away, heterozygous k-mers contribute to the 1x peak, but I'm sure you'll take more things away from this. So yes, Masoud, let's think about this process of whole genome sequencing. We'll start with some individual. This is the individual's genome here. It's very long. In humans, it's more than 3 gigabase pairs. And we have 2 of these. And then we do next generation sequencing. So we sequence this at 50x coverage. But how does this genome sequencing work? Well, you don't go from one end to the other end. Rather, you split the DNA in small pieces, and then you sequence parts of these small pieces. So you get sequencing reads. And then here comes the k-mer method. Then you split those sequencing reads into k-mers. And then we form our spectrum. We count how often is each of these individual k-mers seen. And if you have a sequencing coverage of 50x, then most k-mers are observed around 50 times. But most individuals are not genetically identical between their 2 genome copies, so there are heterozygous sites. And every time you make a k-mer that overlaps a heterozygous site here, it is not observed 50 times, it is observed only 25 times. So that's where the second peak comes from. And for every k-mer that is lost to this 50x peak here, 2 k-mers are added to this 25x peak. Does that make sense? Why is that so? Some people are nodding, but not everybody. Okay, so if you have a heterozygous site here, then the k-mers produced from this strand are different to the k-mers produced from that strand. So for every k-mer that is lost due to heterozygosity from this homozygous peak, you will get 2 k-mers added to this heterozygous peak. And then there are usually some contaminations and sequencing errors, and they contribute to this monotonic thing here that goes up the y-axis.
It's the way that the DNA is fragmented of any influence, or I don't know nowadays, is it just not some random thing?
I think these library preparation methods are getting better, but yes, so there is going to be some bias. So if you take your sequencing reads and you map them to your genome reference, you find that usually the coverage is not even for several reasons. One is this fractionation. Yeah.
Maybe go back to the previous slide.
Sure. This one?
Yeah.
Okay.
That is what you just said.
Okay. Okay, so the relative peak sizes here in this k-mer spectrum of a diploid depend on the level of heterozygosity of this individual. And here I call these peaks 50x and 25x, but more generally, it's better to call them 2x and 1x, right? Because either k-mer is present in both genomes. Yes?
Does this method cope well with locus duplication?
That's a good question. If there's not too much of it, probably yes. Yeah, that's a good point. I think I'll touch on that later. But it's useful not to talk about this in terms of the sequencing coverage, but more in terms of biology. k-mers here 2x because they're present in both genome copies. Here it's 1x present in one genome copy, or you might call them the homozygous and the heterozygous peak. And now with our newly gained intuition about every k-mer lost from this, 2 are added here, we can also come up with a mathematical model that allows us to estimate heterozygosity from looking at these peak heights. So these peak heights, or this heterozygous peak here, will be higher the more heterozygous sites there are in your genome. It will also be higher if you use longer k-mers. If you imagine you have a k-mer of 21 base pairs, then there will be 21 base pairs, there will be 21 k-mers that overlap my heterozygous site. But if you take longer k-mers of 30 base pairs, you'll find 30 k-mers that overlap a heterozygous site. So your choice of k also influences the shape of the k-mer spectrum. Okay, so this is diploids, and really many organisms are diploid, but there are also other ploidy levels. There are tetraploids and hexaploids and whatnot, and there are different flavors of these polyploids. Polyploids can be allopolyploid or autopolyploid. And that's what we look into next. So if you have more than 2 genome copies, there are different kinds of heterozygosity possible. So here are our 4 genome copies, and you may have the situation like as shown here at the top, where all 4 copies are identical. Or you may have a type of heterozygosity where 1 genome copy is different and the other 3 are identical. Or you can have a 50-50 situation, or you can have 3 alleles, or with 4 genomes, you can even have 4 alleles at 1 locus. So you can then take these different configurations and you can tabulate to what peaks of the k-mer spectrum these would contribute to. If you have a tetraploid, your k-mer spectrum can have 4 peaks, 4 main peaks, and you can make a table. This configuration here, 4 identical ones, the homozygous case in the tetraploid, these will contribute 1 count to the 4x peak.
Sorry, Hannes, this question is very simple. Do we not care about the ordering?
The order of the genomes.
Yeah, in the heterozygous. So, for example, in the case of 50-50, they come like every other, they come together.
Are you talking about linkage disequilibrium?
No, like just saying in your picture, so the middle case where
yeah, you can't tell that from the k-mer spectrum, but there can be, it can give you a strong suspicion. We'll get to that. We'll get to that. So then you can go through these different kinds of heterozygosity here. If you have 3 to 1, you get 1 count contributed to this first peak, 1 count contributed to this third peak. If you have 4 different alleles, you get 4 different k-mers, so you get 4 counts added to this k-mer peak here. And then, well, we need to know how likely it is to observe all these, or each of these different configurations. And there I built on a framework that's called the blockwise site frequency spectrum framework. And that allows you to estimate exactly how often you see these configurations. And these expressions all depend on a parameter that's called theta. So theta is a very basic population genetics parameter. It's called the population scaled mutation rate. So in a model population that is panmictic, there is no assortative mating. Theta dictates the level of genetic diversity, of nucleotide diversity. And so these here give us probabilities to observe each of these different configurations. And then you can multiply these values in along the rows, and then you can sum along the columns, and then you get to some expressions for the, so these are not counts, but these are essentially densities, or relative heights, or relative numbers of k-mers in each of these peaks in the k-mer spectrum. And then you can turn this into a k-mer spectrum and try out different parameters. And so you can see if theta is low, 1/10, then genetic diversity is low, then you expect fewer heterozygous sites. And so what you can see in the plot here, when theta is low, you get this blue curve where the homozygous, the 4x peak is the highest. But if you have higher levels of genetic diversity, like in yellow, orange, or in green, you can see that this peak goes lower, and the first peak, the heterozygous peak, where you have all different alleles contributing, for instance, gets higher. Okay, now we have a model what the k-mer spectrum looks like under certain conditions, but of course it would be nice to flip this around and say, well, if I look at the k-mer spectrum, what are these parameters? So first, let's look at an empirical example. This is a tetraploid plant, and here is its k-mer spectrum. You can see a very prominent peak here, the 2x peak. You can see two shallower peaks, the 1x and the 4x peak, and there is no discernible 3x peak here. Then we can plot our 4 expressions for the relative amounts of k-mers in these peaks, and we can just do a sanity check. Because I don't know if you noticed, but looking at this here, this first peak is always higher than the second peak. No matter what is theta, for each value of theta, the first peak is always higher than the second peak. And what we see here is quite different. The second peak is very pronounced. And then you can ask, well, what values of theta would allow you to have such a configuration? So under which conditions can this second peak be greater than the first peak? Is that the right way around? No. But it turns out, so after some algebra, you get to this expression, and then you can see that actually this inequality here, this part here being less than 0, that's impossible if theta is positive. But the theta is a measure of the mutation rate, right? You can't have negative mutation rates. So something is fishy with this model here. For some of you, it may be obvious. Do you have an idea what might be going on here?
Recent genome duplication.
Okay. Yes. Well, certainly a genome duplication. There are different flavors of polyploids, right? You can have polyploids where the 4, if you have a tetraploid, where the 4 copies are essentially equivalent, like you have in potato. These are called autopolyploids. You have 4 copies, they all randomly cross over and segregate. But then there are other polyploids where there's some divergence between pairs of genomes, right? So this is polyploid because at some point, perhaps a hybridization event happened, 2 diverged genomes came together, then the species doubled up its chromosome count, and you have 2 pairs here between which there is an old homology. So we call these 2 groups here homeologous groups. That's quite commonly seen in many plants. For instance, bread wheat is an allohexaploid. It's got 3 of these pairs of genomes. And so then intuitively it makes sense that we get this big peak here because there will be many sites which are identical in these 2 genomes and identical in those 2 genomes, but they will differ between the 2 of them, right? And so there'll be lots of k-mers that are observed twice, both in this genome and in that genome. And that's why these allotetraploids give you this funny 2x peak. And using this blockwise side frequency spectrum method, you can also come up with expressions for these peaks. And it doesn't matter so much what they look like, you just plug them in a computer. But the important point here is that these now depend on 2 parameters. There is still our theta, the population scaled mutation rate. There's another parameter, t, and that is a measure of the divergence between the subgenomes. Okay, and now we have come up with some models. We can look into applications. It would now be very nice to just take a k-mer spectrum and ask, well, what is the genetic diversity? What is the divergence between subgenomes? What is the ploidy level? And you can do this with a shiny app that is called k-mer. And that allows you to choose between different models of polyploidy or ploidy. You can start with diploids, you can have triploids or tetraploids, and you can have allotetraploids, which have corresponding genomes, no, auto-polyploids, which have equivalent genomes, or also allo-polyploids that have different diverged genomes, where the assumption is that recombination happens only in these homologous groups, but not across groups. And you can see here, k-mer allows you to visualize your empirical k-mer spectrum. That's shown here as a dotted data row. And then in red is shown your fit. And there's both a manual mode that's switched on at the moment, but you can also go to an auto-fit mode where it tries to find parameters that make it match. And this should be a video, so I'm trying to start this. Ah, yes, there you go. So auto-fit now, it's snapped, the fit snapped to the data, and then you can fiddle around with parameter ranges. You can see now it's essentially found it. So this prominent peak here is the 2x peak. There's a 1x peak and a 4x peak. And at the end, you can export your fit. And this is actually a slightly older video. There's now an additional button that allows you to export these parameter estimates. Okay. And so here what I talked about is this allo tetraploid with 2 diverged genome copies. And the estimates for this specific example is that the population scale mutation rate for each subgenome is 0.036. This estimate is for each k-mer. If you want to have an estimate per nucleotide, you divide by the k-mer length. In this case, the k-mer length was 27. So that leaves you with a level of nucleotide diversity of about 1 in 1,000, between 1 and 2 in 1,000, which is something normally observed in plants. It also gives you a divergence estimate between these 2 subgenomes, and that is scaled by the effective population size, NE. So if you have a mutation rate estimate, you can then figure out what is NE, and you can turn this here into an actual number of years. But irrespective of whether you know this or not, you can also look into the divergence between the subgenomes. So that's this small d value, which here is 1.37, and that's again on the level of the k-mer. If you divide by the k-mer length, you get the nucleotide estimate. So here we see that the subgenomes in that species, Euphrasia arctica, have 5% nucleotide divergence, which is quite a lot. And again, if you have a mutation rate, you can estimate how long it's taken for these to diverge so much. Okay, so, but there's no reason why these 4 genome copies have to be in the same individual. You can also come up with situations where you have 2 genome copies, but they're in different individuals. And then you can do a similar thing, although you can arrange your k-mer spectrum in 2D, and you get these peaks here, a landscape of peaks, and you can fit parameters to these. And again, there are some expressions for how many k-mers you expect in each of these. And again, they just depend on t, the divergence time, and theta, the population scale mutation rate. So here's sorry, these are butterfly species. It's just something that I worked on before. So you get estimates of their genetic diversity and estimates for the divergence between them. And as you can see, in many species of insects, they have actually quite large effective population sizes. And so even though they've been diverging for a long time, millions of years, the divergence time estimate scaled by population size is quite small, just 2 times the effective population size. But you don't have to take individuals from different species and say what's the divergence between them. You can also pick individuals from the same population. And by saying they are from the same population, there's obviously no divergence time between them. And so you can reinterpret, or you can estimate instead of divergence time, you can estimate inbreeding in your population. You can do that pairwise, or you can do that pairwise for multiple individuals, so get you a more robust estimate of inbreeding. Yeah, I feel it's gotten a bit quiet here, and I don't know if I lost you or is it.
And just from the application, so what you say is most of the k-mer model relevance as in organisms without reference genomes?
Yes, and it is relatively quick, right? So it may be faster than mapping your data to,
Okay.
if it's whole genome data. So always you need whole genome data. So it's probably quite useful for quality control as well. You may have a bunch of data sets, you've forgotten the labels, trying to make sense of them, things like that.
And some people are aligning reads against genome using k-mer
Yes.
approach. That's much faster than aligning to sort of a linear reference, trying to call it out.
So if you map the read to a genome reference, then you need to allow for mismatches. You need to allow for the situation where maybe the read, parts of the read map in different places and things like that. So mapping a read of 150 base pairs properly is quite a lot of work compared to saying, I have a k-mer, it's 21 base pairs, find me a perfect match. If there's no perfect match, we just throw it away. So using k-mer methods is also a way of dealing with sequencing errors, for instance.
Again, what's the impact of structural variance?
We don't know so well. I will touch on it later a bit. No, it's fine. Hope you'll not be disappointed.
So how much the final parameter estimation depends on the choice of k?
I don't know if we know that very well, but that's something that I'm interested in. And we'll also come back to this. Okay, so this is parameter estimation from k-mer spectra. One other thing you can do, and that's essentially model 3, is comparing sequencing libraries. If you have two sequencing libraries made from the same individual, you'd expect them to contain the same k-mers. But sequencing libraries may differ for many reasons. They may have contaminations in them. They may have been prepped with different kits. One may be PCA-free, one may be PCA-based. If you have a PCA-based sequencing library, you get PCR. You get PCR duplicates. These are a form of pseudo-replication, so you want to get rid of them, but they will affect the shape of the k-mer spectrum. So often it's interesting to compare sequencing libraries, be it for quality control or to actually compare individuals. And now we need to broaden our view of the k-mer spectrum. So far, we've been talking about this monoploid or diploid or haploid and diploid peaks, at least in the diploid individuals. And we have cut off this k-mer spectrum here after this diploid peak. But actually, you can plot out the whole k-mer spectrum, and many of you will know that eukaryotic genomes, they contain lots of repetitive elements usually. So there'll be k-mers that are present at much higher copy numbers. If you visualize this, it looks like this. So our two peaks, the heterozygous and homozygous peak here, they shrink together and they're visible here. But there are many k-mers present at much higher multiplicities, often going into the millions. And there are two scales shown here. In black, that's the multiplicity, the k-mer counts taken at face value. And shown in gray here is these values divided by the sequencing depth. So you can see here heterozygous peak. You see these are k-mers present once. Homozygous peak, k-mers present twice. K-mers back here are things that are present 1,000 times. And that's quite common. If you have transposable elements, you may have thousands or hundreds of thousands. I don't know, tens of thousands of copies in a genome. And if you want to compare these spectra between individuals, you need to bin them, because otherwise it's really hard to align them, you know, if you have counts which are integers. So you can divide by the sequencing depth and then make bins. And it's useful. You may have noticed this axis here, the x-axis is a logarithmic scale. It's useful to make bins that increase in size as you're going to the right. So the breaks between the bins are on the logarithmic scale. And so then what's shown here in gray again is the genomic copy number 1 and 2 and 1,000. What's showing in black here is the bin number that I've just come up with. There's nothing special about this, but in my classification here, these things that are there 1x are in bin number 8, things that are there 2 times are in bin number 15, things that are there 1,000 times are in bin number 18. And these bin scaled k-mer spectra you can compare between individuals and between sequencing libraries. So why might k-mer spectra differ between individuals? Well, you may have 2 individuals of the same species, but they differ in heterozygosity. That's what is shown here. The black individual has a higher heterozygosity, so it's got a 1x peak here. It's not really a peak, but there's a shoulder there. And the 2x peak here is a bit lower than of that gray individual. The gray individual has a low heterozygosity, so it's got no peak here at the 1x copy number, but it's got more k-mers in this 2x peak. And then if you want to compare things, you can, for instance, subtract one of the other. And if you subtract counts in these bins, then you get the gray data line here. If you subtract here the gray one minus the black one, you get some negative counts in this area because the black one has more heterozygous k-mers. And again, if you subtract the gray one minus the black one, you get some positive values here because the gray one has more k-mers that are homozygous than this individual with the black data line. So you get this wiggle here. But then you can also take the cumulative sum of these differences, and you see that after this wiggle, there is no net differences between k-mer counts. So this nicely shows that just heterozygosity doesn't change the amount of nucleotides that are in your genome. You get some k-mers that contribute to this 1x peak and to the 2x peak, but overall the numbers don't change. Now, if you have a partial duplication, even if both individuals are fully homozygous, they only have this 2x peak here, no heterozygous k-mers. If you have a partial duplication, then in one individual, some sequences are there at 4 times the copy number. So these are diploids. If you have a duplication, then you have 4x. And here we can do the same thing. We take the difference between these spectra, so gray minus black, and that's shown here in gray. And so here you can see this little hump because the individual shown in gray has some k-mers here that are not present. And then if you compute the cumulative sum of this, as you go to the right, you can see that there is an overall difference. There is an overall difference in genome size. So the individual shown in gray here has a higher genome size. And you can show this in these comparisons. Then there's something else that might be going on, and that's a technical thing with library preparation. If you have a PCR-based library and you get this pseudo-replication of duplicated reads, then the peaks in your k-mer spectrum get wider. So you get some excess variance in these peaks. And that can also cause wiggling in your comparisons, even though heterozygosity may be exactly identical between these 2 samples. So the amount of PCR duplicates or duplicates in the sequencing data are something we need to be aware of. Okay, but then you can actually use this and look at actual data. So here I use the genome in the bottle data set, which is a wonderful benchmarking data set. There are 2 trios, one Ashkenazi and one Chinese trio. And these 2 sets of 3 people have been sequenced with lots of different technologies and to high coverage. And so you can, for instance, and that's the logo of the genome in the bottle data set. And so you can, for instance, compare what do BacBio data look like compared to Illumina data. And this is like a rainbow. So at the end of the rainbow, there's blue and purple. This is the highest density. And at the start of the rainbow, there's red and yellow. And that's the lowest density. And as you might expect between these two sequencing data sets, most k-mers have similar copy numbers. So most of them align along this diagonal line here. And these are humans. Humans don't have huge levels of heterozygosity usually. And so, as you might expect, most of the k-mers are actually here in purple in this 2x peak. But there are also k-mers at other copy numbers. But overall, largely, the PacBio and the Illumina sequencing libraries of the same DNA look largely identical. But there's something odd going on here. There's this additional spur. So there seem to be a class of k-mers that are present in a certain copy number in Illumina sequencing reads more often than in PacBio sequencing reads. And I've not looked into this, what this is, but this is presumably interesting. You'd want to know what this is. Is it related to library preparations? Has it got to do with sequencing primers? Has it got to do with the way the DNA is fractionated? So this is a comparison of two libraries of the same species and individual. You can also compare individuals from different species. These here are from the same genus. And so in these comparisons, you can show both differences in heterozygosity and differences in repeat content. So you see here the 1x and 2x peaks. And so for this individual here along the x-axis, you can see it's got much blue at 2, but not as much blue at copy number 1. So this is a more homozygous individual. Whereas this individual here on the y-axis got some k-mers also at copy number 1. So this individual here is a more heterozygous individual. And overall, these two species differ in the genome size. One of them has about 1 gigabase pair, the other one has 1.2 gigabase pairs. And so you can see that this pattern here deviates from the diagonal line. This diagonal black line here indicates where x and y are identical, but you can see that along the y-axis here, this individual has k-mers that lie above the diagonal line. So k-mers present at a certain copy number here are present at a higher copy number in that individual shown along the y-axis. And then you can also hone in on specific patterns. For instance, there's this blob thing here, something present at the higher copy number in this individual. And if you do this, you find out that this here is actually the genes of the ribosomal DNA arrays. They have hundreds to thousands of copies, and they can easily change in copy number, even within a species. But they contribute here to genome size differences between the two. So this is a way of comparing genome content, mainly focusing on repeats. But again, you show repeats and heterozygosity in the same plot. Yes, I mentioned that library duplication rate. If you think about how a k-mer spectrum shows you counts, then you might intuitively think, okay, so these peaks of the k-mer spectrum, they should follow the density curve of a Poisson distribution. And if you simulate data, that works very well. But if you look at actual data, you see there's always over dispersions. These peaks are always wider than Poisson distribution. And so what's done, people who fit parameters to these spectra, they use a negative binomial distribution, which you can make wider. And so my TETMAR does that as well, and it fits a parameter to how much over dispersion there is. But actually, you can translate this parameter into the degree of sequencing duplication in the library. And I'd argue that's a useful thing to know. So that's going to be a new feature of TETMAR, that it will tell you the degree of sequence duplication in your library. And here is another animation. Oh, yes. Okay, so the more sequence duplication you get, the flatter these peaks become. And if you have too much duplication, you can't actually discern these different peaks. And the wider each peak becomes, they also attract each other, right? They move closer together. So then it becomes really hard to fit any parameter to these spectra. So that's a problem to k-mer spectrum analysis, but it's also annoying if you do the traditional thing of mapping these reads to a reference, because your counts, there's so much uncertainty around counts. So it's always good to use PCR-free libraries or to try to remove PCR and other duplicates from sequencing data. Okay, and here come the structural differences. I touched on this previously, where I said there was a duplication. But I'm really interested in figuring out whether we can actually look at k-mer spectra and tell how many indels are there in the genome. And I think it should be possible, because indels contribute differently to these heterozygosity patterns to what SNPs do. So here, I'm sorry, this is a bit blurry. I copied that from a book. Here, what you can see is how the shape of a k-mer spectrum changes if you change the k-mer length. So let's start with green. K-mer length k equals 21. There is the homozygous peak here, and there's the heterozygous peak here. And as you increase the k-mer length, you go to k equals 25. Relatively speaking, the diploid peak, the homozygous peak goes down, and the heterozygous peak goes up. There's another thing that you observe here, which is that this whole thing shifts to the left. For you, it's this way. And that is because if you have a sequencing read and you turn it into k-mers, the longer your k-mers, the fewer you get out of it, right? If a read is 150 k-mers, 150 nucleotides, and you make a k-mer of length 150, well, then you get only one. Whereas if you have a length of 10, you get 140 out of it. So what this does is, as you increase your k-mer size, these peaks get higher, heterozygous peak gets higher, but also things shift to the left. But there is a predictable relationship between the heights of these two peaks. Because as I said, every k-mer, every SNP that's overlapped by a k-mer produces new k-mers, and the longer your k-mers are, the more of these you get. Okay, but if a k-mer overlaps an indel, that's different. If you have an indel, say a deletion of 50 base pairs, then you have a gap in one strand, and you have 50 base pairs present in the other strand. So there'll be about 50 k-mers that are contributed to the heterozygous peak. But there aren't an equivalent 50 from the other strand. And so if you modify your k-mer length and look how this pattern changes, this change should deviate from your SNP-only expectation, if that makes sense, right? So the rule is, for every k-mer that goes away from this peak because of a SNP, you get 2 added here. So with this expectation, you can say what's going to be the shape of these different colored lines, but this will not match if you have indels. So, and this mismatch, I hope, can be exploited to estimate how much structural variation, at least presence-absence variation, there is in genomes. Okay, so that's, I guess, the most sophisticated thing. So now we've reached the summary. Whole genome datasets can be summarized as k-mer spectra, and these k-mer spectra can be used to estimate heterozygosity, ploidy, other library features, and hopefully, I think, also the amount of indels present in an individual. K-mer analyses are a form of genome profiling. And what is genome profiling? It's the assessment of genome properties in absence of a genome assembly. And yeah, there you see a spectrum with a diploid and a heterozygous peak. And I hope this was useful to you guys.
Thanks, Hannes. Do you have a question?
The methodology to optimize the selection of k-mer length you to optimize the prediction of the theta and the t parameters for specific data.
Well, you want them to be longer. I think it's more this. technical considerations mainly, not these theoretical ones, but there are technical limits. So one thing is you want your k-mers long enough so you don't by chance get identical ones. The other thing is the longer you make your k-mers, the bigger these data structures become where you store them. So usually what people use is 21 or 27. But it's right, if you have an individual that's got a very low heterozygosity, you may want to choose a longer k-mer length just to capture more of these heterozygous SNPs, so that your heterozygous peak turns into an appreciable peak. So one thing that I didn't mention much about is contamination here. This contamination peak may well overlap with your heterozygous peak, and then it becomes really hard to say anything about heterozygosity. But changing k-mer sizes can help you deal with this.
Very quick question. So you put out the plot, is it the TETMA? Is it the app that's showing
Yes.
to export the parameters based on, you sort of move the parameters yourself to put the curve over in front of it. Have you thought of like maybe optimizing that in a way of, I guess, finding parameters based on, you know, distances and things like that?
Yes, so it's tricky. So it uses Optim and R at the moment, and what it does is it computes differences between each point and it sums them up. It's complicated because it's not just one distribution. So there is somehow an underlying negative binomial, but then you have these 4 peaks, and you can think about mixture models and what. But I've not come up with a sort of way, for instance, of getting a likelihood, which would be nice. But if you have any ideas or anybody else, I'd be really interested to hear about this.
Can you use the k-mer spectrum to exclude contaminations, basically? So what's the confidence looking at this spectrum that everything below 50 is errors and contaminations? And if, because you said earlier also it could just,
there is going to be some overlap, right? There are some good k-mers
yeah.
that are just by chance their low copy number.
And would it, but if you would say let's say put a cutoff of 25 or something, and then do you get any use out of it removing the contaminations? Or also you could use the k-mer spectrum and the multiplicity to determine potential contaminations in your
yes,
maybe you don't have the same. Yeah,
absolutely. Different assembly methods work differently, right? Some of them look at k-mers and they will avoid those low copy number k-mers, others won't.
okay.
And that, I guess that's a way of looking at it. You have your assembly, you ask all the sequences in there, how often do I observe them? I actually have some extra slides. There you go. So one thing that people like to do is this completeness plot. So here the outline of this k-mer spectrum is the one of the tetraploid that I showed you before. So there is a very high 2x peak and there's a shallower 4x peak. And then the outline of this k-mer spectrum comes from the sequencing reads, but the color filling it in tells you how often are these k-mers present in the genome assembly that we have. And this is a genome assembly of an allotetraploid. There are 2 diverged subgenomes. So you'd expect that things that are observed, k-mers that are present in only 1 subgenome, in this 2x peak here, they should be present in your assembly only once, because they're either in 1 subgenome or in the other subgenome. And that's what you can see here. Sorry, it's so small. Red means it's present in your assembly, in your reference, once. But this purple color here means these sequences are present in your assembly twice, which makes sense. So these are k-mers that are observed, that would be number 4. And so it makes sense that they are present in both subgenomes as well. So this is about how often they are seen, but you can also tell which k-mers are not present. So this here is a different assembly, and you can tell this assembly here is worse because many k-mers in this 2x peak are actually absent. They are black. They are absent from this assembly. So this is a way of comparing your sequencing data set and your genome assembly and making sure it's complete and k-mers are present at a sensible copy number. I feel that's not exactly an answer to your question, but maybe relevant enough. Different interest. Was that enough stuff on structural variation?
Okay, if we don't have any other questions, I want to say thanks again on this.
Thank you.